Hey YouTube, how's it going? YakScience here with another biology video. Today we're going to be doing an introduction to Mendelian genetics. So who was Grigor Mendel? Grigor Mendel is considered by many to be the father of genetics, and he's given that prestigious title because he was the first person to really scientifically and experimentally address the question of how are genetic traits passed down from generation to generation. That's a question that had baffled scientists even before him, because people noticed that offspring tended to look like their parents, and yet no one could really scientifically address that question until Mendel. And so he addressed that question doing his famous experiments with pea plants, but before we get into those, let's just go over some really important basic definitions. So what are alleles? Alleles are variant versions of a gene, or different versions of a gene. And what we mean by that is, let's say we have a gene for eye color. An example of an allele for eye color might be a genetic sequence that codes for blue eyes, or a genetic sequence that codes for green eyes, or brown eyes, right? You get the picture. So those are variant versions of a single gene. Now, alleles could be dominant or recessive, or traits could be dominant or recessive. You could think of it in either way. But a dominant means that you only require one allele in order to express the trait corresponding to that allele. Okay? So in other words, your genetic makeup is a combination of your mother's genome and your father's genome, where half of your genome is attributed to your father and half to your mother. And so your mother and father will each give you an allele for a given gene. So if in the case of a dominant trait, you only need one, right? So if your mom gave it to you, that's all you need. You don't need both. Whereas with a recessive trait, you need both recessive alleles in order to express that trait. Okay, so you need two copies. Mom and dad both have to give you a recessive allele in order to express that trait, like eye color, for instance. Okay, so a genotype can be understood as a set of alleles, right? Because you get one from mom and one from dad, the combination of those two alleles is your genotype. Okay, genotypes can be homozygous or heterozygous. In a homozygous genotype, both alleles will be the same, whereas in a heterozygous genotype, the alleles will be different. You'll have maybe an allele for green eyes from your mom, but an allele for blue eyes from your dad. Okay, and finally, the phenotype. Phenotype, you can understand it as being the physical expression of your genotype, or the physical appearance, which is kind of a basic way to think of it, but the physical appearance that results from your genotype. So going back to the eye example, the actual color of your iris would correspond to a phenotype. That's an example of a phenotype. Okay, so now that we have these definitions out of the way, let's talk about Mendel and his experiments. Okay, so before we talk about the experiment itself, we should note that Mendel used pea plants for his experiments, and one really important characteristic of pea plants is that they could both self-fertilize and cross-fertilize. In other words, you could transfer pollen from one flower to the ovule of another flower and make them have offspring, make them have cute little flower babies together. Or you could self-fertilize, which means transfer pollen from one flower to the ovule of that same flower and make it have offspring with itself. So Mendel started his experiment by isolating two pure breeding lines. A pure breeding line is essentially, you could think of it as an organism where if you self-fertilize it, right? We just talked about self-fertilization. If you self-fertilize it, all of the offspring in every single generation would have that exact same phenotype as the parent, no change. So a pure breeding tall line would have all tall plants, regardless of how many times you self-fertilize this parent, right? Every single generation would be tall. Similarly, with a pure breeding short line, every single generation will be short, no changes. Okay, so that was step one, isolating two pure breeding lines, and that he called the P generation, or the parental generation. So what did he do next? He actually crossed a pure breeding tall plant with a pure breeding short plant. And what he noticed in the next generation, really interestingly, is that it was 100% made of tall plants. Okay, there were no short plants in this next generation, which he called the F1 generation. Then what did he do? He actually self-crossed the F1 generation with itself, or self-fertilized it. So I'm going to write self over here. And the next generation was even more interesting than the F1 generation Because the short phenotype reappeared, which was super fascinating to him, right? There was no short phenotype here in the F1, and yet it reappeared in the F2. And specifically, he noticed that there were 787 tall plants and 277 short plants. So not only did the short plants reappear in this F2 generation seemingly out of nowhere, but he kept on doing experiments like this, and he kept on seeing a very similar ratio in all of these experiments where you would have a roughly 3-to-1 ratio. Okay, that's a really important point, that in this type of sequence of crosses, you will get a 3-to-1 ratio of the dominant phenotype to the recessive phenotype, right? He called, by the way, the short, in this case, the recessive phenotype, because it was hidden in one generation and then reappeared in another generation. That's a very typical characteristic of recessive traits. Remember back in the beginning of the video, we talked about what a recessive allele is. This is an example of it, right? It was nowhere to be found here, and yet all of a sudden it appeared here. So short was recessive and tall was dominant. So when Mendel would carry out similar experiments, he would notice a 3-to-1 ratio of the dominant phenotype, like tall or yellow color or rough seeds, whatever trait he was looking at, a 3-to-1 ratio of the dominant to the recessive phenotype. So he looked at this and he came up with this theory for how this all worked. So now that we know about DNA and genes, let's talk about this 3-to-1 ratio in terms of genotypes. So if we were to give this a genotype, what would it be? Given that it's a dominant trait, as we proved here, this would definitely be homozygous dominant. So let's just use the letter A for simplicity. So the genotype of the pure breeding tall plant in the P generation would be big A, big A. And similarly, the pure breeding short plant would be little a, right? Now, if we know that we crossed these two and we got 100% tall, what would be the genotype of all of these individuals in the F1 generation? Could it be big A, big A? The answer is no, because you get one allele from mom and one allele from dad. Whichever one is mom and dad, I'm not sure. So the genotype of all of the F1 individuals would be heterozygous, big A, little a. Okay? And finally, when we self-cross these, we get a 3-to-1 ratio of dominant to recessive phenotype. So let's take a look at what a cross of a heterozygote would look like, also called a monohybrid cross. So if we were to look at a Punnett square showing the cross between big A, little a, and big A, little a, look at this, look at the ratios we get. What would be the phenotype of this big A, big A? It would be tall. What about this big A, little a? It would also be tall. Remember, because we talked about dominant alleles only requiring one copy, right? This only has one big A, but we know because it's dominant, it'll express the tall phenotype. So tall, 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 short. So notice there's 3 talls for every 1 short, a 3-to-1 ratio. And this matched up so perfectly with this that Mendel was able to come up with these really important genetic rules, these rules governing basic genetics. One, of course, being the existence of discrete heritable factors, which later came to be called genes, right? He didn't know what a gene was, so he called it a discrete heritable factor, but his intuition was there. The idea that there's this entity that's actually passed down from generation to generation became clear through his pea plant experiments. And then next was the principle of segregation, which was this idea that 2 alleles actually separate from one another and get placed into gametes, okay? So basically when we saw the P generation, big A, and little a, each of those parents had gametes that had one of those. Of course, both of them are the same, big A, so each of the P generation's gametes would have a single big A allele. So segregation, principle of segregation, is saying that for any combination of 2 alleles in your cells, when it comes to producing gametes, they will separate from one another and end up in separate gametes. They'll segregate from one another. And finally, equal segregation, which is sort of a subset of the principle of segregation, says that the gene pairs distribute equally into the different gametes. And then there's this concept of random fertilization, which basically states that the gametes from each parent will fuse to form a zygote randomly. And so that's a basic introduction to Mendelian genetics. I hope that clarified some stuff, and thanks for watching.